Welcome to Advanced Concepts and Perspectives of Membrane Physics, an introduction. For roughly the first 30 years, two big questions guided the field. First, can the structure and the physiological functions of multi-component cell membranes be explained by classical thermodynamics of mixtures? Second, does phase separation, modulated by specific lipid-protein interactions, organize functional domains in membranes? At the same time, Bending Elasticity Theories for Soft Shells by Wolfgang Helfrich and Evan Evans revealed that many vital cell functions depend on the membrane's bending elasticity. Here is the core idea behind bending elasticity. A membrane vesicle resists bending with an energy cost proportional to curvature squared. A compact summary is E equals 2 kappa times the integral of mean curvature squared, plus Gaussian terms. These ideas, developed by Helfrich and Evans, motivated high-precision measurements of bending moduli in vesicles and red blood cells, building on theories by Brochard and Safran. Now, let's visualize phase separation and so-called lipid rafts. We draw a bilayer patch with two coexisting regions. On the left, a fluid phase made mainly of glycerol lipids. On the right, a condensed phase enriched in sphingomyelin and cholesterol. We label the fluid L-alpha phase and the condensed liquid ordered phase, L-alpha C. Because the two phases have different packing and thickness, a line tension forms at the boundary. Extrinsic proteins with polybasic domains can be recruited by electrostatic coupling to the cytoplasmic leaflet, while specific hydrophobic matching helps integral proteins partition into the best-fitting domain. How can we summarize the thermodynamics of mixtures observed in many membranes? First, mammalian phospholipids can be approximated as a quasi-binary mixture, glycerol lipids versus sphingomyelin including gangliosides. Sphingomyelin has long, mostly saturated chains and transitions near body temperature. Glycerol lipids have shorter, more unsaturated chains and melt near 0 degrees Celsius, forming a fluid L-alpha phase. Second, cholesterol prefers saturated chains and fluidizes solids, forming a dense liquid ordered solution together with sphingomyelin, the L-alpha C phase. Hydrophobic matching can sort proteins between organelles. Consider two bilayers of different hydrophobic thickness, like plasma membrane and Golgi. A protein has a fixed hydrophobic length. If the mismatch is about plus or minus 0.5 nanometers, Monte Carlo studies estimate an insertion cost near 2K BT per lipid, and around 20K BT in total for some proteins such as band 3. At equilibrium, a shell of roughly 30 length adapted lipids accumulates around the protein. This favors partitioning into domains or organelles with a better thickness match. One illustrative example is transferrin receptor-mediated iron uptake. At the plasma membrane, transferrin receptors bind iron-loaded transferrin. A vesicle buds off, then fuses with early endosomes. Under acidic conditions, iron dissociates. The endosomal membrane can segregate into a sphingomyelin cholesterol-rich domain containing the receptor and a glycerol lipid-rich domain. Rab4 GT passe then routes the receptor-rich domain directly back to the plasma membrane, bypassing multilamellar bodies. To summarize, coexisting fluid and condensed phases form through mixture thermodynamics, with cholesterol as a key modulator. Bending elasticity, described by a curvature energy, governs shape, adhesion, Perling instabilities, and trafficking. Hydrophobic matching and electrostatics recruit proteins to domains and organelles, which in turn guides cellular logistics like the transferrin receptor pathway. These concepts together build a physical picture of how membranes organize and function.